Okay, so welcome everybody. Our speaker today is Hannah Small. Hannah is a PhD student in the Department of Cognitive Science at Johns Hopkins, working with Dr. Leila Ajik. And she received her bachelor degree at the University of Richmond and then worked as a technical associate in Dr. Eve Frederico's lab at MIT. Hannah is currently interested in studying the spatial organization of the brain in the context of human social perception. She's also the recipient of a 2023 NSF Graduate Research Fellowship. And today she's going to talk about one of her projects, an investigation into simultaneous spatial and linguistic processing during a naturalistic movie. Now, so without further ado, please join me and welcome Hannah Small.
Hi, thank you so much for inviting me to give this talk. I'm really excited to share some of this ongoing work. Um, it is, we're like currently writing it up for a paper, but it's still in progress. So if you have any comments or feedback, I would love to hear your thoughts. Um, and Kelly let me know that this is a more informal setting. So, and most of my work involves Sherlock. So this is kind of themed in a detective like case briefing way. So you'll have to bear with me for the theme for the rest of the presentation. So here we go with the social, visual, and language processing during a naturalistic movie. Oh, let me get that started. Okay, so here's our case briefing. Um, Social cognition depends on integrating information from both vision and language. In our daily lives, we see what's happening around us, including social interactions, and then we also hear what's being said in those social interactions. Um, and from previous literature, we know that bilateral superior temporal sulcus supports many parts of dynamic social processing. For example, there are regions that process dynamic faces, voices, theory of mind, language, and biological motion all along the STS. And this research primarily used separate controlled stimuli to look at each of these functions. And in this work, we're going to be looking at social interaction and language areas and investigate how they support the processing of a naturalistic stimulus where there is simultaneous visual and verbal signals. Okay. And so a little bit of background on visual social interaction perception. Previous work has found in like posterior STS, a region that is selective for social interaction. And the way this region is found is using a contrast of interacting point light dyads versus non-interacting point light dyads. I'll show you some example stimuli in a bit, but in A on this slide, that is showing a group map of a bunch of participants and showing this contrast. And typically, this is a right lateralized region. So this is just showing in the right hemisphere. And then in B, you can see an individual brain and this individual's other regions that are nearby. And so in blue, there's the theory of mind region. And then we have the face, the STS face region in yellow, and then motion region in green. And you can see that the red social interaction perception region is distinct from these other nearby regions that could also be involved in social processing. Other recent work from my lab that I'm in is that using a bunch of naturalistic 3-second videos and an encoding model approach, a graduate student who actually just defended her dissertation, Emily McMahon, she showed that in the social interaction region, it's particularly sensitive to communicative actions. So you can see that there's a picture of what a communicative action would look like. Um, and Also, there's background on language processing. Verbal communication is processed by this language network, which is the system of left lateralized regions that are strongly selective for language processing over non-linguistic inputs and tasks. And here I'm showing a probabilistic map of over 800 subjects and the contrast of a comprehensible language input and a non-comprehensible language input. And you can see in the left STS in this yellow region, that's where the most overlap is happening between subjects. Right in the STS, there is a lot of language processing. And then also in B, we see individual subjects. And you can see that in most subjects, this network is left lateralized and compared to the right lateralized social interaction region. Oops. Oh no, I have to go through all my animations. Here's a review of everything I just said. And so in this work, we're asking the questions, are social interaction and language regions distinct? And how are they processing these simultaneous visual and linguistic signals in a naturalistic stimulus? So first, we're going to locate our suspects, and that is really just selecting our regions of interest using some contrasts. To find the language regions, we use a contrast of intact speech versus degraded speech. And so here is an example of intact speech.
I would say I'm a cat person, even though most cat people are so strange. People that admit to being cat people are always so bizarre. Like,
And then here is some degraded speech. And so we take the contrast of this, and then we take the top 100 most selective voxels across the left and right STS. And for this particular subject, we see that they are mostly in the left hemisphere. And then to find the social interaction regions, we use a contrast of interacting. So here's an example of interacting point light figures. And this is what was used in the original study that found this region. And then here's some non-interacting point light figures. And again, we use this contrast to find the social interaction regions. And again, taking the top 100 most selective voxels over the left and right STS. And so here in this subject, these voxels are mostly right lateralized. And there is very little overlap between the language voxels and the social interaction voxels. And we can quantify the overlap between these two regions using the dice coefficient and then averaging across all of the subjects. And we can see that there's very little overlap between these two regions across all the subjects, with an average of 0.05 in the left and less than 0.01 in the right. And this is consistent with different threshold, like different number of voxels that we can choose. There's not much overlap between these two regions. Another measure we can look at is the hemispheric distribution of these voxels. And so here I'm showing the proportion of the top 100 most selective voxels that fall in the right hemisphere. So values close to 1 mean that most of the voxels are in the right STS, and then values close to 0 means that most of the voxels are in the left STS. And for social interaction, we can see that for most subjects, most of the voxels are in the right STS. And for language, it's the opposite pattern, where most of the voxels in subjects are in the left STS. And then also we can verify the suspect identities by looking at their responses to conditions in held out data. So for the social interaction localizer, we are asking if the voxels that we found using 2 of the runs also show selectivity in the third run. And so here I'm showing the responses to interacting point lights in purple, and then non-interacting point lights in a more pink color. And you can see that in both the left and the right STS, social interaction STS regions, there is social selectivity. And then we can also do the same for the language regions. And so the response to intact speech is shown in orange, and then the response to degraded speech is shown in yellow. And you can see that in both the left and right, we see a language selective response. And then the next thing we can do is look at within each of these regions, what are their responses to the other experiment's conditions? So in other words, are the social interaction selective voxels language selective? And here I'm showing the language responses. You can see that there is some selectivity for the language region where there's more response to intact speech compared to degraded speech, but it's not nearly as much response as in the language regions. And then the language selective regions are not selective for social interactions. Yeah. And so our interim results are to the question of, are these social interaction language regions distinct? Spatially, yes, and both display the typical opposite lateralization and are mostly non-overlapping. And then functionally, social interaction regions have some selectivity for language. So a little less distinct functionally. And so next, now that we have our regions, we want to look at their responses to simultaneous visual and verbal signals. And to do this, we will look at their responses to a naturalistic stimulus, particularly the first episode of the BBC series Sherlock. And here is just a little example clip that I pulled out.
It's an old friend of mine, John Watson.
Afghanistan or Iraq?
Sorry?
Which was it, Afghanistan or Iraq?
Afghanistan, sorry. How did you?
Molly, coffee, thank you.
Okay, that was a quick little thing. And then we labeled different features of the movie to link the features to neural responses. And all of these that I'm describing right now are human annotated. So we have the presence of faces in the frame, or sorry, in the TR, since we're going to be showing this in fMRI. And speaking, whether there's speaking occurring in the TR, whether there's a social interaction, the number of agents in the scene, valence of it, arousal, and there's a couple more. There's going to be a full list on another slide, but feel free to ask any questions about this. And so we have all of these features for all of the TRs of the movie, which is every 1.5 seconds in this case. We also automatically extract some features using deep neural networks. And first, I'll talk about the visual features that we extract. Put very simply, a visual deep neural network learns to transform pixel input of an image into a label. For example, this network learns through modifying the weights between the different neurons in the hidden layers that this image contains a coffee. And the activations within the hidden layers contain this neural network's representations of that image and representations that support this task that the network was trained on, which is categorization. And we take these activations within these hidden layers, and then we use that to predict neural responses. So previous work has shown that when you're comparing a convolutional neural network, specifically AlexNet, which is the same network that we will be using in this work, the different layers going up the network correspond to higher and higher levels of the visual hierarchy. So early layers of AlexNet correspond to V1, and later layers could correlate with VTC. And we also use neural networks to extract features of the spoken content of the movie. So these networks learn to predict a masked word. So in this case, the network would learn to predict the word cup. And again, the hidden layers are what contain the representations that support this ability. So we will be using these activations throughout these hidden layers to predict responses in fMRI to the spoken content of the movie. And in particular, we're using a language model called BERT, which is a bidirectional transformer that builds up a contextual representation of the words And then we also are going to be using a word level embedding called word2vec. But just this process of using language model representations to predict responses to language content in fMRI is becoming very popular, and they're typically used to predict neural responses to listening to language. There's one very popular data set that is 20 hours of listening to podcasts. And so they extract the representations of all the spoken content of this podcast, and then they predict the fMRI responses to those same words using neural networks. And but typically there's no visual input in those cases. So we're using this method to look at the simultaneous visual and verbal signals using vision and language neural networks. And this map is kind of confusing to look at if you're not used to flat maps, but the band of red, like towards the bottom of the brain, that's the like temporal lobe. And so that's just showing a high encoding performance of a particular language model. In this case, it's a very, very large language model. Okay, so here's the full pipeline for getting the movie features. We first get the first frame of every TR. So here's just a couple example frames. And we feed that through a visual neural network to get the activations of all of the layers of the hidden layers. And we are also extracting some motion energy using a motion energy model called PyMoten, which uses pyramids of spatiotemporal Gabor filters to extract motion energy. And then we also get the transcript of the movie and timestamp all of the words. In this example, the timestamps correspond to the end of the sentence, but we have the timestamps for every single word. And then we feed those sentences through a sentence BERT. So getting those sentence contextual representations. And then we also feed it through a word2vec model, like I mentioned earlier, to get word level embeddings. And then we have all of this is the list of the full perceptual features and then the social affective features that we have. And one problem that some people might have realized is that we're taking, since we're having the first frame of every TR, our features are all sampled at the same sampling rate for like the visual features. But for language, that might not be the same sampling rate as the fMRI signal. So we have to downsample those features, and we use that. We do Lanczos interpolation for that, and here's just a little schematic of how that occurs. It's a very simplified schematic where each word has a spike at the time at which it occurs in the stimulus, and then there's a Lanczos kernel that it gets convolved with, and then that gives us a continuous signal that we can sample at each TR. And then that is the feature that we use to predict the fMRI signal. And if people have questions about that, I can answer questions later. So before we predict the brain activity with all these features, it's useful to get a sense of how similar all of these are to each other. Typically in naturalistic stimuli, things are all correlated. So here we use canonical correlation analysis to measure the similarity between all of our feature spaces. And this is a method of just getting the similarity between feature spaces of any dimension. And it finds linear combinations of each feature space that are maximally correlated with each other. And so here you can see in the top left of the matrix that is all of the AlexNet layers and how similar they are to all of the other AlexNet layers. You can see they're all very similar to each other, which is what we would expect. And then for the language model that's in the bottom right corner, all of the layers are similar to each other. But then in the bottom left corner, we see that AlexNet and SBERT are very weakly correlated with each other throughout the movie, as measured by canonical correlation analysis. So we also see that visual features are correlated with valence. which is not surprising since this is a production that was directed by someone in order to elicit like emotional responses from people. So it's not surprising that the visual features of the movie correspond to valence. And we also see that linguistic features are correlated with the most social features that we have, including whether there's a social interaction occurring, speaking, whether there's like mentalizing about another character, for example. And that's also not surprising because in a movie and also in like daily life, when there's the social interaction occurring, typically the people are speaking. And so these are correlations that we can't really get away from when using naturalistic stimuli. But importantly, the visual and the language response, the features are not very similar. And to link these features to voxel-wise activity, we will be using linear regression, specifically banded ridge regression. And so here I'm just showing that we are showing the visual part of the episode and also the spoken content. These both happen simultaneously and they get processed by a particular person's brain. And we are extracting the voxel-wise bold time response to the simultaneous visual and verbal signals. And then we have all of the features of the movie, and each feature is duplicated at multiple time delays. And this is to account for different hemodynamic delays across cortex. And then we learn beta weights for each of those feature spaces in order to predict the fMRI bold time series. And the banded ridge regression comes in when we are learning these beta weights. This is all done in a cross-validated way, and we learn some hyperparameters in an inner loop of the cross-validation that scale each beta. And that helps deal with overfitting due to like very high dimensional spaces like neural networks. They have a very high dimensional space compared to the one-dimensional features. And the hyperparameter will scale all of the weights of one particular layer, for example, by the same amount. And then these hyperparameters also can deal with correlated feature spaces, because if there's redundant feature spaces, then the hyperparameter can scale one down, and it learns that in a cross-validated way. So then we get our performance score of the encoding model by calculating the explained variance, or the coefficient of determination, which is big R squared. And then that is what we are going to be looking at next. So here's a group map of the encoding model performance. And you can see that it performs well across cortex. And we can also see that it performs well within our regions of interest that we're looking at. And in the purple bars, I'm just showing the intersubject correlation of these regions. And you can see that our encoding model is explaining more variance than an intersubject correlation can, suggesting that it's fitting some subject-specific information that intersubject correlation can't access. And finally, we can look at which features are contributing to this predictivity. To answer this, we are looking at the product measure, which shows the predictive contribution of each feature space. And this takes the correlation structure of the feature spaces into account. And so here, I'll just walk through what this measure is. So here, I'm just showing all of our features again, and then the beta weights that are learned. And the beta weights are a measure of the importance of each feature in the model. And then the correlation of each feature with the actual time series in the test set. So this is on data that was not used to train those beta weights. We get the correlation with the actual time series to capture the correlation structure of all of these feature spaces. And then you take the product of these two to get the product measure. And so, for example, to get the social interaction product measure, you take the beta weight for social interaction, feature space, and then the correlation of that feature space with the actual time series, and you get the product measure. And so importantly, this is calculated on held out data. So it's a test of how it generalizes, but also how important that feature is in explaining the data. So here I'm just showing a voxel-wise map of one particular subject, and these are the features that we would be looking at, and I'm going to color each voxel according to the feature that explains the voxel the best in terms of this product measure. So whichever feature has the highest product measure, it'll be colored blue for AlexNet, purple for motion, orange for SentenceBERT, and white for Word2Vec. And so here are the results for this subject. And you can see that visual features in blue are predicting the best in most of cortex, including the ventral stream and also into higher regions. But you can also see the orange is the SentenceBERT and white is Word2Vec, and those are showing up in regions that we would expect language regions to be. And then we can also look at the product measure of each feature within the social interaction and language regions to see which features contribute to explaining their responses. And we see that visual features explain about 60% of the total variance in the social interaction regions, and then about 40% of the total variance explained in the language regions. And then the rest of the variance is explained by language features in both the social interaction and language regions. So this is showing us that embeddings from both the visual and the language networks explain variance in both social interaction and language regions, even though these features are they're weakly correlated. So, and we're also accounting for any correlations using the banded ridge regression. And so this is showing that both of these regions seem to have multimodal responses. So even language regions are sensitive to what is being seen at any given time, the visual content of the scene. And social interaction regions are sensitive to what is being said in the movie as well. So sensitive to vision and also what's being said. And we haven't done much analysis on it, but another interesting thing that I've noticed is that the social interaction regions, the SentenceBERT in particular, does well compared to Word2Vec. And in the language regions, Word2Vec is doing much better. It's like on the same level as SentenceBERT. So it seems that in particular in the social interaction regions, the contextualized sentence representations seem to be what are helping support the social interaction regions. Okay. And we can also look at how well just the human annotated one-dimensional features do. They kind of get drowned out by the very high-dimensional neural network activations, but here I've just limited to looking at the preference map, but only using the one-dimensional features. And in green is the social feature, and you can see that in the STS, it's doing pretty well. And then also in like a more salmon color, that is the speaking feature. So that is also doing well across STS. And in the dark green is the face feature. And you can see there's a nice little face region in the ventral stream for this particular subject. And so the final verdict for this is, are social interaction language regions distinct? Spatially, yes. Functionally, kind of. And how are they processing the simultaneous visual and linguistic signals? Both regions are well explained by these mostly non-correlated visual and linguistic signals in the movie, suggesting that they are multimodal regions, which has been shown before, but not shown in this way. But also that so we're showing that we need to be studying these regions using multimodal approaches in context. And this is just one framework for doing that using neural network activations and an encoding model approach. So that's the main verdict of this work. I have a little bit of extra work that is more in progress, and it seems like I have some time, so I'll present those too. So the other thing we can do with this data is select voxels using the naturalistic movie. So instead of using the controlled stimulus, instead use the features of the encoding model and how well they predict neural responses. So here I'm just showing the top 100 voxels that show the largest product measure for the social feature, and then sentence BERT plus the word2vec feature to estimate what the language features should be. And so this is just in this particular subject, you can see that they have these voxels occur in both the left and the right STS. And if we compare the controlled stimuli to the naturalistic stimuli, we can see that in the controlled, we see again the right lateralization of social interaction and the left lateralization of language. But in the naturalistic stimuli, we see that it's more bilateral in this case for both social and language. And then we can also look at the responses of each of these regions to the controlled stimuli to see if they are selective in any way. So again, here's just this is the results I already presented. So the social interaction regions are social selective and language selective. The language selective regions are just language selective. And if we select the voxels using the naturalistic movie, if we use the social feature on the left, we see that we're picking up social selective voxels, but that are also language selective. And in the on the right, we see that when we use the sentence BERT and word2vec feature to select the voxels, we're picking up language selective voxels. That may also be social selective, but the responses are very, very low in the right. So this is kind of a cool verification of our results. The social feature ends up picking up very social selective voxels according to the controlled stimuli. And then we can also look at the overlap, the spatial overlap between each of our localizers, the language localizer, which I'm going to be showing in red, and the social interaction localizer, which I'm showing in yellow, to the voxels that are picked out with the social feature in the movie, which will be in blue. And so here you can see it's kind of hard to see, very preliminary work, but in the right hemisphere in the green, you can see some green voxels, which are the overlap between the social interaction localizer and the social feature. So there's just some overlap only in the right STS, and it looks like around mid STS. And then we can do the same thing, but switching out that blue feature to be the language feature. So a sentence BERT and word2vec. And so now the overlap will be in purple because it'll be overlapping with the language localizer. And you can see in the left hemisphere, there's some overlap between those two regions. If we look across subjects, if we just quantify this again using the dice coefficient and then average across subjects, we see that on average, there's very little overlap between these regions, but there is a little bit of a correspondence where the social feature from the movie overlaps more with the social interaction controlled localizer than it does with the controlled language localizer. Very small numbers though, but that is really only in the right STS. In the left STS, the social feature overlaps with both the social interaction and the language controlled localizers. And then there's a similar pattern for the language where the sentence BERT and word2vec feature of the movie, those voxels overlap with the language localizer, and not very much with the social interaction localizer, and that's in both the left and the right STS. But yeah, that's a little bit more preliminary work. But so I'll go back to this slide. And yeah, that's all I have for you today. And thank you for your attention, and I'm happy to take any questions.
Hey, Hannah, thanks so much. That was a really great talk. I think to start, there's a question in the chat from Jin. Have you tried to use visual stimuli to define language ROIs? Curious if that's going to affect the overlap.
Oh yeah, no. So we only have the intact versus degraded speech localizer right now. We chose that one because we are collecting this in neurotypical and also autistic participants, and we wanted to use a localizer that is accessible for everybody, and that is like the easiest one to use. So it's more of a logistical reason why we picked that one.
Yeah,
But that
makes
would be really
sense.
easy.
Also, just to confirm, you guys are working in adults, right?
Yeah, this is all in adults.
Cool. I'll open it up, guys. Anyone questions you want to come up and ask? Well, you think, don't worry, I have a question. Hannah, this was really great. The work is super cool. I was wondering how on a couple of your group maps, it seems like there's a particular part of the temporal lobe, like the anterior inferior, that doesn't seem like it's modeled well by any of the things that you're trying. And I didn't know if you thought about that or if you had any thoughts. It's a little far from the STS, but I thought it was interesting.
I
Yeah,
see.
yeah, so in the encoding model, and I feel like the other group map, it was kind of similar, that one chunk, and I wonder if maybe it's like a signal to noise or if you think there's like a more interesting reason.
Yeah, I think that area is tricky for us, I've noticed. I think it's always a problem, but in particular, I forgot to mention this is all within an intersubject correlation mask. So the encoding model it wasn't run in that region because there wasn't intersubject correlation there. But that could be because of the signal to noise rate problem. Yeah.
Yeah, that makes sense. And then, so I know you said you guys are working on writing this up right now. I was curious, are you guys trying to include like the bonus slide you added at the end of trying to define things from the movie and then looking back? Because I thought that that was really cool, but also maybe could be something in and of itself, like kind of a separate project.
It's that's good feedback. It's unclear. Well, we're still trying to figure out our story for the paper. So
Yeah.
that's good feedback to hear.
Yeah, I'm doing some similar things in kids, but just looking at like theory of mind and language. So I'll shoot you an email because your results seem pretty similar to what we're getting to in development. So we can chat a little bit offline about that.
Yeah, that would be awesome. I think I've seen some of because Zainab came and gave a talk,
Oh
and I was
yeah,
like,
she came and gave a talk to your lab. Yeah.
yeah, and I was looking on the website. I think are you Kelly?
Yeah. Sorry, I
Okay.
realize that you can't see me. You just see like the room. Yeah, I'm Kelly.
So I looked at your poster looking at the theory of mind and language regions, and I was like, oh my gosh, this is really relevant. So
Yeah.
I'll be looking forward to your email.
Yeah, it seems similar. Okay, anyone else last questions before Hannah and I just chat? Okay, Hannah, let's thank our speaker one more time. Thank you so much. That was really great.
Thank you.
All right, guys, feel free to use the last of our time to